and welcome to the Vaxart Fiscal Year 2020 Corporate Update Conference Call and Webinar. At this time, all participants are in a listen-only mode. A question and answer session will follow the formal presentation. If anyone should require operator assistance during the conference, please press star zero on your telephone keypad. As a reminder, this conference is being recorded. I would now like to turn the conference over to your host, Mr. David Holmes of LifeSci Advisors. Please go ahead, sir.
Thank you, Hector. Good afternoon and welcome to the Vaxart Corporate Update Conference Call. With me today are Andre Florio, Vaxart's Chief Executive Officer, and Sean Tucker, the company's Chief Scientific Officer. During the course of the conference call, we will be making forward-looking statements regarding the future events and the future performance of the company. These forward-looking statements may be accompanied by such words as should, believe, could, potential, will, expected, plan, and other words and terms of similar meaning. Examples of such statements include, but are not limited to, statements relating to our ability to develop and commercialize our product candidates and clinical results and trial data, our expectations with respect to the important advantages we believe our oral vaccine platform can offer over injectable alternatives, expectations regarding our ability to develop effective vaccines against new and emerging variant strains, the expected role of mucosal immunity in blocking transmission of COVID-19, and our expectations with respect to the effectiveness of our product candidates, including our potential role in mitigating the impact of COVID-19. All of these statements are based on the beliefs and expectations of management as of today. These statements involve certain assumptions of risks and uncertainties and could cause actual results to differ materially. We assume no obligation to revise or update forward-looking statements, whether as a result of new information, future events, or otherwise. Investors should read carefully the risks and uncertainties described within the Safe Harbor section of our website, as well as the risk factors included in the company's most recent quarterly and annual filings with the U.S. Securities and Exchange Commission. Following Vaxart's prepared remarks, there will be a question and answer session, and a replay will be posted on the investor section of the company's website. At this time, I would like to turn the call over to Andre Florio. Andre, please go ahead.
Thank you, David. Good afternoon, everyone, and thank you for joining us. I'm excited to provide an update on our clinical programs and outline some of our priorities for the coming year. We started last year with a peer-reviewed publication in the Lancet ID featuring results from a phase 2 clinical trial of our pandemic flu vaccine, a trial that was sponsored by BARDA. In this flu challenge study, we showed that our oral tablet vaccine protected against flu, as well as the leading injectable flu vaccine, Fluzone. Importantly, our oral tablet flu vaccine produced much lower levels of serum neutralizing antibodies than Fluzone, less than 1/10 as much, but was just as protective. This is a very important finding. In the context of COVID-19, serum neutralizing antibodies are seen as a key metric for injectable COVID-19 vaccines. So it is very important to recognize that for our oral tablet vaccine, serum neutralizing antibodies are not an appropriate proxy for efficacy. The protection of our oral vaccine in the flu challenge study was driven in part by B cells that recognize HA of IgA class, as opposed to HAI titers for the injectable flu vaccine. These results provide important evidence that our vaccine is working through a different mechanism of action than injectable vaccine. In fact, our oral vaccine generated a broader adaptive immune response, including mucosal T cells and local IgA, to effectively protect from influenza. Early last year, at the beginning of the COVID-19 pandemic, our team had the foresight to design a vaccine that incorporates both the spike S and nucleocapsid and viral protein. This approach was based on our expectation that coronavirus was likely to mutate and spawn new S mutant variants. The N protein is more conserved, less prone to mutations, and also a great target for potent T cell responses. This and recent studies suggest that targeting the N protein may induce broader protection from the new variants of SARS-CoV-2. Therefore, we are very excited about moving towards phase 2 with a COVID-19 vaccine candidate that could be transformational, an oral tablet that works through a very different mechanism and which could prove to be a very valuable globally against the fight against, in the fight against coronavirus. We believe our COVID-19 vaccine constructs have several important advantages. One, the triggering of mucosal immunity, the first line of defense against airborne pathogens such as coronavirus. Two, the tablet. And three, the simplicity and lower cost of distribution of a room temperature stable pill. Recently, we have seen the emergence of new SARS-CoV-2 strains against which some of the leading injectable vaccines offer reduced protection. At the same time, it has become clear that mass vaccinations by needle take a long time and that new strains may emerge faster than we can vaccinate people. Therefore, a better solution is needed, given that COVID-19 may be a challenge for years to come. Our strategy for tackling this global challenge is to employ a multivalent portfolio approach, looking to improve on many aspects of the first-generation injectable vaccines, including convenience and speed of administration, breadth and depth of protection, and ease and cost of distribution. This involves advancing our S and N vaccine candidate into phase 2, while in parallel developing S-only vaccine constructs. We believe that with our COVID-19 vaccine portfolio, Vaxart will have multiple paths forward, depending on geography. In the developed world, annual boosting of those previously vaccinated or infected, with the potential of conferring cross-variant protection. In the developing world, de novo vaccinations with a much more practical solution than cold chain dependent injectables. Based on the fundamental underpinnings of our oral delivery platform, we have also restarted our norovirus program. As a quick reminder, the United States sees approximately 20 million norovirus cases every year. Health economic impact is felt mainly by children under the age of 5, as well as elderly over the age of 65. Recently published health economic data from the American Journal of Preventive Medicine puts the cost at over $10 billion annually. Based on this data and internal estimates, we believe there is an annual $10 billion plus opportunity for a norovirus vaccine in the US alone. We are currently in the clinic with this norovirus vaccine, administering booster doses to a few patients who participated in our earlier norovirus phase 1 bivalent study. The data we generate here should give us an indication of the duration of protection provided by our vaccine, as well as the magnitude of response that is generated by the booster. In addition, this year we have 2 additional studies planned. The first will evaluate our norovirus vaccine in people over the age of 65, and the second will be a challenge study that will provide us with efficacy data in this disease model. I'll conclude by reminding everyone that to date, we have dosed nearly 500 subjects with vaccines based on our oral platform across 13 clinical trials targeting 7 different viruses. And now, to provide some additional scientific background, I would like to pass the microphone to Sean to discuss our COVID-19 oral tablet vaccine phase 1 results. Sean?
Thanks, Andre. As this was a phase 1 study, the primary objective was to determine the safety of VXA-CoV-2-1, our oral COVID-19 vaccine delivered by tablet. Overall, the vaccine appeared to be well tolerated. 60% of subjects reported no symptoms in the 7 days post-vaccination, and among those 40% who did experience symptoms, the majority had only mild symptoms. These resolved without the need for medical treatment. No subjects discontinued the study due to a solicited adverse event, and to date, there have been no serious adverse events reported. The secondary objective of the study was to determine immunogenicity of the vaccine. Overall, immune responses against SARS-CoV-2 were observed in approximately 85% of subjects. We are very encouraged by the substantial T-cell responses we saw as a result of the tablet vaccine, and I'm delighted to say that these results are the best we've ever seen in a human study. The cytotoxic T cell responses at day 8 had a high percentage of cells that made gamma interferon, TNF-alpha, and CD107A in response to stimulation with the S protein, with substantial increases compared to the first day of the study. Potent T cell responses may offer protection against severe COVID-19 illness across multiple variants. B cell plasmablasts, which are the immune cells involved in antibody production, also increased in subjects post-immunization. There was also upregulation of the mucosal homing receptor and surface IgA on those B cells in a dose-dependent manner. While no neutralizing antibodies responses were observed in the serum of subjects, preliminary analysis showed that the increases in IgA responses to the S protein, receptor binding domain, and/or the N protein could be found in the majority of subjects. IgA, as a reminder, is a class of antibody that plays a crucial role in mucosal immunity, the first line of defense against respiratory pathogens. These IgA antibodies, specific IgA antibodies, were detected in several different compartments, including nasal and saliva samples. Given the dose-dependent manner in which the B cells of interest were activated, future studies of this candidate will focus on dose ranging and boosting to increase the mucosal immune responses to SARS-CoV-2. In 2019, we anticipate starting several studies, which were highlighted by Andre before. For COVID-19, we are going to perform a phase 2 study, Part A, immunogenicity and dose ranging, Part B, efficacy, and a boosting study, previously vaccinated and infected subjects. For norovirus, we are currently in a phase 1B trial for booster vaccinations at 12 months or longer post-first dose, and we are planning dose-ranging phase 1 study in the elderly, 65 years of age and older, as well as performing a norovirus challenge study. We look forward to advancing multiple studies throughout the year and delivering both clinical data and publications to support a proprietary oral vaccine platform technology. We expect these data throughout the year to provide the medical community and investors with a great opportunity for this novel and groundbreaking approach. With that, I will now ask the operator to open up for questions.
Thank you. At this time, we'll be conducting a question and answer session. If you would like to ask a question, please press star 1 on your telephone keypad. A confirmation tone will indicate your line is in the question queue. You may press star 2 if you would like to remove your question from the queue. For participants using speaker equipment, it may be necessary to pick up your handset before pressing the star keys. One moment, please, while we poll for questions. Your first question comes from the line of Mayank Mamtani with B. Riley Securities. Please proceed with your question.
Thanks, team, for taking my questions and appreciate the update. So maybe just picking on the last topic that, John, you talked about around the study design. Can you give us a little more color on the number of patients, the timelines, and maybe also in context of the recent FDA guidance document that came out last week, how do you sort of accommodate for the focus there being on neutralizing antibodies?
Hello, sorry. Can you hear me now? So, let me answer.
Thank you.
Let me answer that question. So the first thought, of course, is the Part A is a dose-ranging study where there's going to be 3 different dose levels and priming and boosting at all 3 levels. The N's are 16, but there's also, it's broken out into elderly and young people to sort of get a good mix. And that guidance will basically be able to provide us insights into how to go forward with a efficacy study and take the proper dose and dosing regimen into a phase 2 efficacy study, which we plan to run outside the U.S. and placebo-controlled trial. And I believe the N's for that study were approximately a total N of about 900. And obviously, with the recent guidance that has come out with the FDA in terms of using potentially surrogates, we have to reevaluate that. And certainly, from this candidate, the VXA-CoV-2-1, if it's a T-cell approach, we wouldn't be able to use a surrogate from the standpoint of proving essentially efficacy. We would still have to do a efficacy study. and prove that the vaccine can prevent something such as severe illness and hospitalization.
And Mayank, this is Andre. I just wanted to add to that our conversations with regulators indicated that they are fully aware that our vaccine is a mucosal vaccine, and they indicated flexibility in working with us to find a path to approval. So we don't expect the same kind of metrics and proxies that are being developed for each of the vaccines to apply to us necessarily.
Actually, that brings me to the second question that I had around, yes, your, T-cell responses, comparably superior to what we have seen from others. And, the recent Nature paper from Sterling, and there's a lot of, literature coming out highlighting the importance of IgA also. Maybe, T-cell comparison has sort of started to come out against other peers, but can you comment on how your IgA responses are looking like, relative to other, more of the intramuscular vaccines? Do you have any thoughts on that?
So just, as a highlight, I would say that our IgA responses in terms of when we've compared, in different indications head to head, our, mucosal approach definitely produces much more mucosal IgA than a intramuscular approach. I mean, that's pretty clear. I mean, and in this study, we have not, of course, done a head-to-head comparison between our approach versus an injected vaccine. But I would say that, as far as I know, we're the first vaccine company to report getting mucosal IgA of significance. So as far as I know, that's where we're at.
Okay. And my final question was on norovirus. Do you have any plans to do pediatrics, since that is also a big part of the population that gets exposed to it, and also what does that mean as a read through, because pediatric and COVID is also a very big need.
Yeah, I think that right now that. Oh, sorry. Go ahead.
No, you take it, Sean.
So yes, we will be moving into the pediatric space on norovirus. I would expect this to happen in a number of years rather than this year. So I don't think it will really overlap. Well, we hope it won't overlap with COVID too much. The key thing for us is that while right now all of our vaccines are delivered in tablets, which are really easy to swallow for someone who's 12 and older, it's not, a format that will allow a small infant to basically take it up, or a child. And so we're developing other formulations to allow it to essentially be swallowed in a sort of a slurry with a buffer. So we're working on that. But we will be moving into the pediatric space on norovirus.
Okay, understood. And just last question then. Do you expect coronavirus vaccine to be a single boost or a prime boost with a 1 month, or you just don't know? You have to run the study. You're not sure yet.
Well, obviously, the answer is different with people that have been priorly infected or people that have been priorly immunized than it would be with people that are naive. We are doing this dose ranging study, of course, with people that are naive, so we can understand more about the kinetics. But you might expect that, for a lot of the Western, world countries, a lot of people will already be immunized, within a year, and we'll probably look at this in terms of a one-dose sort of regimen on a boost.
Got it. Thanks so much for taking my question. And Mayank, just to elaborate a little bit on that, I think what we want to do is to develop a portfolio approach that can accommodate different needs in different geographies, and also keep pace with the evolution of the virus, right? So whereas last year the challenge was to develop any vaccine against one strain, I think the challenge is now becoming developing vaccines that can offer a broad protection against various variants that are present now, as well as future ones. So that's where we think that vaccines that target S and N perhaps in combination with vaccines that only target S for various strains may play a role.
Yeah, that makes a lot of sense. Thanks, Andre, for that additional clarification.
As a reminder, if you'd like to ask a question, please press star 1 on your telephone keypad. As a reminder, if you'd like to ask a question, please press star 1 on your telephone keypad. One moment, please, while we poll for questions. Your next question comes from the line of Vernon Bernardino with H.C. Wainwright. Please proceed with your question.
Thank you. Hi, guys, Andre and Sean. Thanks for the additional details as far as the update on the programs with the COVID vaccine are concerned. May I basically asked all my questions, but one thing that perhaps we haven't talked about in a while is wondering if you could provide an update, and that is there anything going on with your collaboration with Johnson & Johnson? Thank you.
So that collaboration is still ongoing, and as you can appreciate, both us and J&J have been extremely focused and busy with our respective COVID-19 programs. And once there is something to announce as far as the next steps with our flu programs, we'll make some public announcement.
Have you had any discussion with them as far as collaborating on the oral tablet COVID vaccine development?
Nothing of substance that would announce publicly for now.
Okay. And when again may we see results from the ongoing COVID vaccine trial?
So the only thing we said is that we're looking to start the phase 2 trial in Q2, and as we get closer to that, we'll elaborate on timelines following that.
Okay. Thank you. Look forward to the next progress report.
Thank you so much.
Your next question comes from the line of Shubhendu Senroy with Brookline. Please proceed with your question.
Hi. I'm Shubhendu on behalf of Kumar from Brookline. I really appreciate the business update. Thank you. I had a couple of questions. One was, so for Q2, in terms of enrollment of patients for the COVID vaccine study, do you see any challenges, especially for older patients, now that we have few vaccines out there? Do you see that could be a problem or anything?
Hi, this is Andre. Thank you for your question. Look, it's hard to predict. It's hard to predict the future, but we think that given the attractiveness of an oral COVID-19 vaccine, given the relatively small number of patients that we need for the dose ranging study, and given that we still expect a significant share of the U.S. population to not have been vaccinated, at this point, we don't see enrollment as being a significant issue. But we'll update as necessary as we progress.
Got it. Thank you.
And I would add that I think conducting a placebo-controlled trial in the U.S. and broadly speaking, developed world may be a challenge because it's placebo-controlled and they are large. But in the dose-ranging study, all of the subjects are going to be on active drugs, so that minimizing the challenge.
Right. And in terms of the product needed for phase 2, you think you would have enough supply for that?
Yes.
Okay. I had a couple of questions for the COVID results. And could you please comment on how long is the Cdft response is expected to last? I mean, you have a really good response, Cdft cell response, but then Do we have any idea of how long it is going to last?
Yes, this is Sean, by the way, of course.
Hi, Sean.
We expect the CD8 responses to last a long time, and the reason why we think so is because when people that have been infected with SARS-CoV-1, the original SARS, you can find T-cell responses 17 years later and cross-reactive against SARS-CoV-2. So we do expect that one of the advantages of a T-cell response is that they are long-lasting. We know that the magnitudes of the responses we looked at prime are pretty high, and we know that the convalescent people from SARS-CoV-2 get about a 0.2%, in gamma interferon response. So our expectation is that our responses will stay and be maintained, and we'll be able to find those cells. And when we'll have more guidance probably later on that. But our expectation is that T cells are going to be very long-lasting.
Right. And so in the same breath, you expect the overall protection against illness and infection by the vaccine to expect long as well?
So so I would say, if you're talking about protection based on a sort of a T cell response, we do expect it to be long lasting. A T cell response is not going to by itself will not provide sterilizing protection, but would protect against severe infection and hospitalizations, and that's what we expect to last for quite some time. And again, if we can boost the mucosal responses are really robust, we also potentially can actually block infection.
Gotcha. Thank you so much. Thank you for taking my questions.
No problem.
Your next question is a follow-up from Vernon Bernardino with H.C. Wainwright. Please proceed with your question.
Hi, Andre and Sean. Thanks for taking my follow-up question. Given the challenges of perhaps conducting an optimal investigational COVID vaccine study in the United States, as well as many countries in the Western world where there has already been a rollout of the authorized vaccines, have you considered perhaps trying to do a study with the oral tablet vaccine just in Brazil, for example, because obviously that is a variant of concern and one in which there has been very low vaccine efficacy demonstrated by all the vaccines so far that have completed phase 3 or in phase 3.
So, Vernon, thank you for your question. I'll answer first, and then I'll let Sean add anything if he wants to. So, from the get-go, a month ago, we said that our intention was to conduct larger phase 2 studies, placebo-controlled, looking for efficacy outside of the U.S. So that remains the plan. However, at this point, as we said in the opening remarks, we see the world kind of diverging in terms of need. So in the developed world, there is a lot of interest for one, cross-variant protection, and then boosting, periodic boosting, whether it's annual or so on. So one of the studies that we are looking to do in the developed world is to boost those that have been previously infected or vaccinated, and then to do de novo vaccinations, as you suggested, in developing countries such as Brazil, India, and so on. Sean, I don't know if you want to add anything.
Vernon, your question is spot on. Our intention is to open up regulatory paths in Latin America, South Africa, and potentially India to basically be able to address, as you suggested, the strains that are emerging that are not necessarily covered by the vaccines very well. And again, with this product that drives, a cross-reactive T cell response, that would be a pretty good way to find and look for efficacy.
Terrific. Yeah, I just asked because it seems like it'd be the easiest study to do of all, especially if you're going to go to a boost strategy eventually anyway.
It's a very good question and strategy.
Right. Thank you. Look forward to the continued progress, as I said. Thank you.
Your next question is a follow-up from Mayank Mamtani with B. Riley Securities. Please proceed with your question.
Just on the topic of strategy, I thought I'll ask you this question. What about doing a challenge study? Have you guys, opened any conversations with, certain countries that have shown some willingness to at least be open-minded to it? And then my other higher level question was, how do you sort of see the broader landscape of vaccines, that are looking at either oral modality or going after N protein? Like what sort of, you see broadly among your peers?
So let me take the first question. Sorry, Mayank. Sorry, could you please repeat the first question?
Just potential of doing a challenge study, like something similar to what you did in flu, where you started the call with.
I was thinking about your second question while forgetting the first one. So we've had concrete discussions in some regions where they are open to doing challenge studies, but we feel like it's going to take some time for those to actually become reality. But of course, we'd be very interested in doing that in whatever region where regulators are open to doing that. And as far as your second question, I'll let Sean elaborate. We are not aware of any mucosal or oral vaccines that are, relatively advanced or in the clinic. So we don't think too much about that competitively. Sean, I don't know if you have anything else to add.
Yeah, just on the oral side of things, I'd say that we've been in this business in the oral tablet for a long time, and there's definitely a lot of learning to do to get it to work, and we think our technology is very important for that. In terms of again, nothing has been reported. I think we're the first mucosal vaccine to report data as far as I know. And if competitors come out and report data, that's great. In terms of an N response as well, I think there's only a few other companies that are looking at N, and so far they haven't reported data. So I can't really comment on, what they've shown yet.
Understood. And actually a follow-up that I just got was, on the publication cadence from phase 1 data, like how are you thinking about, that, Sean, just kind of putting this in a peer-reviewed journal, and also I know you've considered working with universities like Stanford, where thinking about this as a heterologous kind of approach to a protein-based vaccine or an mRNA vaccine. Or even looking at studies where you can maybe in a prospective way look at, your data and compare it, be it T-cell, be it IgA, to, some of these other approaches in the more real-world setting. Like, when can we see some of that, more sort of prospective and more, direct comparison that we can, in a proper study, proper controlled study?
Yeah, again, I don't think we can provide guidance on timing for these things, but we certainly will write up results for the phase 1 study. There's definitely some things that we'd like to, finish and, detail out before we actually go and write the manuscript. And you did mention something. Yeah, we do have lots of friends in the academic world, and there's ways for us to basically sort of look at our T-cell responses in a sort of more, what I should say, head-to-head manner by collaborating with some of the sharing samples, for example, and everything else. Some of those things will just take a while to get in place, the agreements, but I do expect that we will get data coming out in Q2 without providing exact guidance at this point. But that would be a reasonable expectation, is that we'll have a lot more to talk about in next quarter.
Sounds great. Looking forward to that. Thanks for taking the follow-up.
Ladies and gentlemen, we have reached the end of the question and answer session, and I would like to turn the call back to Mr. Andrei Floroiu, CEO, for closing remarks.
I want to thank everybody that attended this conference, and we're looking forward to progressing all of our programs and to updating you soon. Thank you so much.
This concludes today's conference. You may disconnect your lines at this time. Thank you for your participation.